Hey students, it's Mr. Lucido. Thank you for taking the time to watch this quick video. In this video, I want to accomplish 4 things. The first thing is I want to give a quick recap of Unit 1 as we prepare to transition into Unit 2. The second thing is I want to talk about 2 big terms in Unit 2 that we're going to use a lot, prokaryotes and eukaryotes. I'm going to distinguish between the 2 of them. The third is I'm going to talk about how those 2 categories are actually connected by something called the endosymbiotic theory. I'm going to talk about the 3 core pieces of evidence that are used in support of this theory. And then fourth, I'm going to highlight some, not all, of the connections between organelles that you've been reading about and you watched in that other video lecture, the connections between organelles and eukaryotic cells, because we want to think about the cell as a system rather than these separate components inside of a cell. So that's what we're going to do in this video. All right, so let's start with just a quick recap of Unit 1, which again was called the chemistry of life. So in it, we learned about just some of the key chemistry topics that we need to understand life at the molecular level. And so one of the first things that we learned about were these 3 different types of bonds: non-polar covalent bonds, polar covalent bonds, and ionic bonds. And remember that the difference being that in covalent bonds, we have the sharing of electrons. When it's a polar covalent bond, that means that there is unequal sharing because the electronegativities are significantly different between the atoms involved. So chloride actually has a really high electronegativity, so it's going to hog those electrons, giving it a partial negative charge in this molecule. Whereas a nonpolar covalent bond is where they're equally shared. I like to think of ionic bonds as kind of like an extreme of polar covalent bonds. It's where you have such a big electronegativity difference that it's actually a complete transfer rather than a sharing of electrons from one atom to the other, giving it a full charge rather than a partial charge. So such as sodium chloride, where chloride has a full negative charge and sodium has a full positive charge. So we use that idea of different types of bonds to then study water. So water has 5 major properties like cohesion, adhesion, expansion upon freezing, high specific heat, and an effective solvent, all because it is able to form these hydrogen bonds between other water molecules. And that's because we see 2 polar covalent bonds present in a water molecule because of the difference in electronegativity between oxygen and hydrogen. And so like we see here, a hydrogen bond is simply where you have a partially positive hydrogen interacting with a partially negative, in this case, oxygen from a different water molecule. And that leads to all of the different properties that we see with water that we highlighted. We also use this idea of bonds and partial charges to think about the other types of molecules in biology. If you remember, there were 4 major types: carbohydrates, lipids, proteins, and nucleic acids. And we spent a little bit of time thinking about the structure and a little bit of their function throughout Unit 1. So over here we have the major example of lipids. And lipids, we are mainly going to be dealing with phospholipids. And phospholipids, the structure is given right here. We can use what we know about charges to actually understand some of its properties. And so phospholipids are known to have a hydrophilic head and a hydrophobic tail. And that's because of the bonds that are present in the tail and the head. All hydrophilic or hydrophobic means is that water is able to or not able to interact with it. Okay, in order for water to interact with a molecule, it has to have a charge. And so in this case, it's a hydrophilic head because you see this little negative charge right there on the oxygen. A hydrogen in a water molecule can form a little hydrogen bond there because, again, hydrogen in water has a partial positive charge. But the thing that happens in the tail is that these are all carbon-hydrogen bonds, right? We talked about how these structures with just the lines are carbon-carbon bonds or carbon-hydrogen bonds. And those are non-polar. And since they're nonpolar, there's no charge, partial or full, for water to interact with. And so because it doesn't interact with water, it's hydrophobic. So those were lipids. Now the other three types of molecules we can think of in terms of monomers and polymers. And so you learned about the two major processes involved with polymers and monomers. Dehydration synthesis, as we see in this figure, is the building of polymers, whereas hydrolysis is the deconstruction of polymers into monomers. This happens to be carbohydrates down here, with glucose monomers being linked together in dehydration synthesis or being broken apart in hydrolysis. All right, and then we thought about nucleic acids, which again, nucleotides are the monomer and nucleic acids are the polymer. And we compared DNA and RNA to think about how they're similar and different. One of the differences was that the 2' carbon in RNA and DNA are different in that RNA contains an oxygen connected to the 2', but in DNA, there is no oxygen connected. And then lastly, we spent a lot of time thinking about the 4 levels of protein structure, primary all the way to quaternary, and thinking about the different types of interactions that occur at those different levels. This happens to be, I think, hemoglobin, demonstrating quaternary structure, which is multiple polypeptides interacting with each other. So again, that was just a very brief overview of Unit 1. But what we're going to start to think about is molecules start to arrange into these larger structures in living organisms called cells. So lipids are a big component of the cell membrane, and the membranes that compartmentalize organelles. DNA obviously is a huge role in this cell, as well as protein. So we're going to start thinking about in Unit 2, how do these molecules, how do their properties connect to what we see as the systems in a cell? All right, so moving right along to our next objective here. So we've done a quick recap of Unit 1. Unit 2, the title again is Cell Structure and Function. And so in this part of the video, I'm going to talk about prokaryotes and eukaryotes, which are two major categories of cells that we're going to see throughout this unit and throughout the year. Now, the first thing is I just want to distinguish between these two by actually looking at what they're called. So prokaryotes and eukaryotes, those words, they actually have a meaning that clues us into what they are referring to. So a prokaryote, that word means before nucleus, before nucleus, as a rough translation. Eukaryotes, that means true nucleus, and this gets at one of the major differences between these two types of cells. But before we go further, that's just the meaning of both of those names. Now, some examples of prokaryotes and eukaryotes. There's two categories of prokaryotes. One that we've heard of, bacteria, and two are archaea. And that's just a separate type of prokaryotes, but those are the two major categories. Eukaryotes, we have things like fungi, plants, and animals, right? So these are our eukaryotic organisms. Okay, so now let's move on to how are these two categories of cells similar and different. So to highlight their similarities, so both cells, prokaryotic and eukaryotic cells, they both contain DNA. So they both contain DNA, which is the type of macromolecule that's needed to store information for building proteins, which both prokaryotes and eukaryotes need. So both of them have DNA. Put this in the middle here. And the second thing that they have in common is that both eukaryotes and prokaryotes have cell membranes. That is just that basic barrier between the inside and the outside of the cell. And we see that illustrated here in the figure. The third major similarity is that both prokaryotes and eukaryotes have these structures called ribosomes, which again are needed alongside the DNA in order to produce proteins from the information stored in DNA, which are needed for just basic cell function. So those are the three major similarities: the DNA, ribosomes, and membranes. Now, there are differences. So there's actually a lot of differences between these two types of cells. Kind of at the surface level, we have, based on their name, in prokaryotes, we don't have a nucleus, right? So there's no nucleus, which is this membrane-bound structure which protects the DNA. So you can kind of see we have DNA in the prokaryote, but there's no membrane surrounding that DNA. But in a eukaryote, the DNA is stored inside the nucleus right here. So the first major difference is that eukaryotes, they have a nucleus, but prokaryotes do not. The second difference is kind of along those lines. Eukaryotes are known for their membrane-bound organelles, and all that means are these structures like mitochondria and the nucleus, the Golgi apparatus, or in plant cells, chloroplasts. And by membrane bound, that just means that these structures are surrounded by a membrane. Okay. But inside of a prokaryote, not only do they not have a nucleus, but we also don't see any membrane-bound organelles. Okay. Now, one thing you might have noticed is, well, what about ribosomes? So ribosomes are not membrane-bound organelles. They are a subcomponent of a cell, but they're mainly made up of actually rRNA and some protein that makes up a ribosome. So they don't actually have a membrane. Okay, so those are the first two differences. I think the third major difference, which is more general, is that eukaryotics tend to be more complex. All right, they tend to be more complex, whereas prokaryotes tend to be a little bit more simple in their functions. And kind of along these lines, typically we see eukaryotes are a little bit larger and prokaryotes are a little bit smaller. Okay, so while there are multiple types of prokaryotes and multiple types of eukaryotes, the differences and similarities that we've highlighted here hopefully give you a little bit of a better idea of how we can distinguish between prokaryotes and eukaryotes. All right, so we're moving right along to our third objective here. We've done a quick recap of Unit 1. We've talked about prokaryotes and eukaryotes. So now I want to talk about this third objective. And so if you remember, one of the things I mentioned in the last segment about prokaryotes and eukaryotes was that typically prokaryotes are smaller and a little bit more simple in their functions in comparison to eukaryotes, which are a little bit larger and a little bit more complex. And so one question that has come out of that observation has been, did eukaryotes evolve in some way from prokaryotes? And what this has led to is this very commonly known theory in the field of biology called the endosymbiotic theory. Okay, the endosymbiotic theory, which is summarized right here in this diagram. Okay, so what does this theory suggest? Well, this theory suggests that at one time there were only prokaryotic cells on Earth, and the precursor to eukaryotic cells, being just a larger prokaryotic cell at the time, through a process known as endocytosis, engulfed another prokaryote, kind of like we see right here in this diagram. So it literally engulfed another prokaryote, which happened to be an aerobic prokaryote. I'll talk about that in a moment. As it engulfed that prokaryote had its own membrane at the time, but now, because it was engulfed, it was surrounded by the membrane of the prokaryote that engulfed it, as we can see right here. Now, the idea is that over time, this cell that had engulfed another prokaryote developed other membrane-bound organelles, such as a nucleus, as well as in plant cells, a photosynthetic prokaryote was engulfed as well. And over time, we saw the complexity of these cells increase such that now we have what we see as eukaryotes. And so that's the idea of the endosymbiotic theory. Now, there are three core pieces of evidence that are presented in this theory that I'm going to introduce in this lesson. So these three pieces of evidence all deal with two organelles found in eukaryotes, mitochondria and chloroplasts. So mitochondria, which we see right here, are the site of cell respiration, which we'll study in Unit 3, which is the conversion of energy from one form of chemical energy into another. But what's observed in mitochondria and in chloroplasts, which are the site of photosynthesis, is that both of these organelles are unique from other organelles in that they have two membranes. They have two membranes. If you look at both of their structure, they have an inner and an outer membrane. We have an inner and an outer membrane. And what that suggests is through this mechanism that I mentioned earlier of engulfing that aerobic prokaryote, which was suggested to be the precursor to mitochondria, and the photosynthetic prokaryote, which is the precursor to chloroplasts. When they were engulfed, they maintained both their original membrane and the membrane of the cell that engulfed it, which is why we have an inner and an outer membrane still with mitochondria and chloroplasts. So that's the first piece of evidence. The second piece of evidence is that when you look at both of these organelles, what's very interesting is that both contain their own DNA. They both contain, it's much smaller than the amount of DNA found inside of the nucleus, but it contains DNA that allow for the production of proteins that are needed within that organelle. Other organelles don't have that, but mitochondria and chloroplasts both have their own DNA that suggests, again, at one time they were independent prokaryotes. All right, and the third piece of evidence is actually what happens when eukaryotic cells divide. Now, if you remember from your biology class, right, when cells divide, we call that mitosis. However, in prokaryotes, it's still division. We call it fission, though. They divide in a slightly different way, but the result is the same, right? We have one cell becoming two. And what's interesting is that when cells, eukaryotes, divide, mitochondria and chloroplasts, they actually go through that fission process that prokaryotes go through. So right before the cell divides, both mitochondria and chloroplasts, if you're talking about a plant cell, or just mitochondria in an animal cell, their DNA will duplicate, as we see over here. Their DNA will duplicate, and then actually they will go through that fission process where it'll split into two mitochondria or two chloroplasts. And so that's very characteristic of a prokaryotic cell. And so that's the third major piece of evidence in support of this theory of endosymbiosis. So again, to summarize those three pieces of evidence, number one is that both mitochondria and chloroplasts have double membranes, an inner and an outer membrane, which suggests being engulfed at one time by a eukaryotic precursor. The second is that they both contain their own genetic information. They both contain DNA. The third is that we see the presence of this fission process where both mitochondria and chloroplasts can independently divide prior to cell division. All right, so before we move on to the next portion, just a quick note about the name endosymbiotic theory. So endo is referring to the process of endocytosis. That was how the cell engulfed that other prokaryote. Symbiotic is referring to the kind of mutually beneficial relationship between the cell that engulfed that prokaryote and the prokaryote that was engulfed, right? For the engulfed prokaryote, they received protection, probably from external stressors, whereas the cell that engulfed the prokaryote received what the in this case mitochondria or chloroplast precursors were able to do, which was either harness energy in the case of a chloroplast or release energy that's usable for the cell, as in the case of the mitochondria. Okay, so to wrap up this lesson, let's talk about this last component. So we're going to highlight some of the connections between organelles in eukaryotic cells. And this is just to get you started on what I hope you continue to do throughout this unit as you study and learn about these different organelles. So in your reading guide, you should have learned just about some of the basic components of these organelles and their functions. But what we want to do is think about the cell, in this case a eukaryotic cell, as a system of these organelles, each contributing to the systems found within the cell. And so the way I like to do this is you think about how the functions are connected to each other. And there is a major flow of information and function throughout a eukaryotic cell that I'm going to highlight, but again, this is only some of the connections that exist between organelles. I encourage you to continue to study how these are connected, but what we know is that kind of where this information flow starts is in the DNA, right? So we have these sections of our DNA that code for proteins. And so what we typically see is transcription occurs in the nucleus of a eukaryote, and the RNA makes its way into the endoplasmic reticulum. Now, this model only shows the rough endoplasmic reticulum, which is a major site of protein synthesis because it has lots of ribosomes. But what tends to happen is that mRNA makes its way into the rough endoplasmic reticulum, where it is translated by the ribosomes embedded in the membrane of the endoplasmic reticulum. From there, that protein that was synthesized is actually sent through a vesicle that kind of comes off of the membrane of the rough ER and moves towards the Golgi apparatus. And the Golgi apparatus will receive that translated protein, and through a variety of processes that happen in the Golgi, you get modifications, you might get additions to the quaternary structure of a protein. There are processes like glycosylation, which is like adding these little sugar tags that help direct where that protein needs to go. But all of those modifications are going to happen in the Golgi, and then the Golgi is actually going to send that protein, a lot of the times, to be secreted outside of the cell, and that will go to either another area of the body or the eukaryotic organism that cell is a part of, but it'll be secreted. Or we'll often see some of those vesicles go elsewhere in the cell. Let's say it was a protein needed in the mitochondria or elsewhere in the cell, it'll be sent there to perform its function. So that's kind of the basic flow of information from the DNA in the nucleus through the rough ER to the Golgi to be secreted or used elsewhere in the cell. Okay, so one last connection to think about here is all of those processes that I highlighted, like transcription and translation and the moving of vesicles from the rough ER to the Golgi, all of those actually require energy, right? Typically in the form of ATP, which again, we're going to learn more about how that is extracted from carbohydrates in the mitochondria, but the mitochondria, right, is supplying the ATP that's needed to allow that flow of information to occur. Okay, now there are other organelles like the lysosome, which contain enzymes to help break down, for example, proteins or other molecules when they're ready to be degraded, that are connected to this major flow of information or other processes in the cell. So as you study, I encourage you to think about not only the function of each organelle, but also how does it connect to other organelles, because that's going to help us to understand eukaryotic cells as a whole. So again, thank you for watching this video. I do hope that you found it helpful as an introduction to Unit 2. Again, please finish that reading guide and watch that boson video about organelles as well. And I look forward to seeing you next week.